282 of the Stem Cell Podcast, Whole Body Regeneration with Dr. Mansi Srivastava. Hello, everybody. This is Dr. Daylon James, and we also have here Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how we can improve and for suggestions on guests. Our guest today is Dr. Mansi Srivastava. She's from Harvard University, and she's on the podcast to talk about her research on regeneration and panther worms, as well as some Evo-Devo stuff. We're going to have a very far-ranging conversation. Looking forward to that. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, interested in generating cells derived from human pluripotent stem cells? Get a free wall chart on the directed differentiation of HPSCs. To learn more, visit stemcell.com/HPSC-differentiation-wall chart. And actually, on that topic, first paper we're going to talk about is a paper looking at the differentiation of iPSCs into corneal epithelial cells for transplant surgery. This is a landmark study coming from Japan. It's a single-arm, open-label, first-in-human interventional study for transplanting these iPSC-derived corneal epithelial cells into the eye for treatment of limbal stem cell deficiency. And you know, this is one of a number of iPS-derived clinical trials coming out over the past years, and we've talked to folks in this field who are working in the area of generating cells from iPSCs for, say, transplantation into the brain, in the case of Blue Rock, and transplantation into other tissues as well. Japan has, of course, taken the forefront in the iPS field, especially when it comes to clinical transplantation, because Dr. Shinya Yamanaka, the guy who first described iPSCs, is, of course, in Japan. And you know, this is again just another example of the great studies that are emerging from Japan in this area. So let's get right into it. Like I mentioned, this is a high-profile paper in Lancet. We typically don't cover papers in Lancet because Lancet is a clinical journal first and foremost, right? So the loss of corneal epithelial stem cells from this area of the eye called the limbus, it's at the edge of the cornea, has some pretty major consequences for vision. I mean, people ultimately can go blind because of this sort of situation. And the pathological manifestations of what you call limbal stem cell deficiency, or LSCD, is pretty tough to treat. And so here, they described, and they said it themselves, the world's first use of corneal epithelial cell sheets derived from human iPSCs to treat limbal stem cell deficiency, or LSCD. And you have to take this into consideration in terms of the study design and everything. This is a non-randomized, single-arm clinical study, which involves 4 eyes of 4 patients with LSCD. So it's pretty small, right? That's just, you know, straight up immediate limitation of the study, but very early study of safety and, you know, potentially somewhat efficacy, and they're going to expand on this a lot more over the years to come. This is done at the Department of Ophthalmology in Osaka University Hospital in Japan. And the 4 patients comprised a woman age 44 years with idiopathic LCSD, patient 1, man age 66 years with a different form of LCSD, which is patient 2, a 72-year-old man with an idiopathic LSCD, patient 3, and a 39-year-old woman with toxic epidermal necrosis. That sounds pretty nasty, I got to tell you, patient 4. And these are allogeneic human iPS-derived corneal epithelial cell sheets. So that's important to note. This is not autologous. You're not deriving iPSCs from the same individuals, from the same patient. But they created these allogeneic human iPS-derived corneal epithelial sheets, what they called iCEPs, and transplanted them into the affected eyes. And this was done sequentially in 2 sets of HLA mismatch surgeries, with patients 1 and 2 receiving a low-dose cyclosporin and patients 3 and 4 not. And like I mentioned at the outset, the primary outcome measure was safety. They looked at adverse events both in the eye and outside the eye. These folks were monitored continuously over the course of 52 weeks, 1 year, and an additional 1 year safety monitoring period. So 2 years at the minimum. They looked at secondary outcomes reflective of efficacy, which were also recorded. And that's very early looking at the efficacy. Primarily, this is a safety study, but ultimately, what they found um, were that there were 26 adverse events during this 52-week follow-up period, which consisted of 18 mild in their description and one moderate event in the treated eyes, and seven mild non-ocular events, off target, not in the eye. Importantly, these are patients enrolled between June of 2019 and November of 2020, so right before or right at the start of the pandemic. And I'm sure you know how tough it must have been to conduct a study of this level during the pandemic, right? Um, and at 52 weeks, they looked at the secondary measures of efficacy and showing that the actual disease stage had improved. There was a correction in distance visual acuity, so they're able to see a little bit better. Um, and this phenotype of corneal opacification, or like kind of the darkening of the cornea, which ultimately leads to the blindness, actually had diminished in all of the treated eyes, all four eyes. Um, and then they followed it up with different analyses of corneal epithelial defects, different symptoms, quality of life, questionnaire scores, which all showed an improvement or unchanged. So no, doesn't seem like there was any negative impacts of the transplantation. And in their words, they felt that the beneficial efficacy outcomes were achieved, in particular for patients 1 and 2. And 3 and 4 also showed somewhat of an improvement, but 1 and 2 were definitely the best in terms of the improvement. And ultimately, this is, again, very early phase. This ICEPS transplantation for LSCD was found to be safe. This is their conclusion throughout the study period of 2 years. And they're going to be doing a larger clinical trial, which is going to further investigate truly the efficacy of the procedure. So, bottom line, this is a safety study, very early safety study, just 4 eyes from 4 people. But hey, promising results, don't you think?
Sure, sure. I mean, it's progress, and I, to be frank, the progress is for me is a bit agonizing, just in terms of how incremental it is, and that's not a negative. It's just necessary, because these cell-based experiments take forever, right? I mean, you're talking about experiments that started before COVID. I mean, wasn't that like 100 years ago? And I'm looking here as you're talking. Lancet, I think, totally merited a Lancet publication. We don't publish. I mean, we don't cover these more clinical, super high profile journals, namely Lancet and New England Journal of Medicine. Those are two big ones. And the last time we covered, I think this might have been before you joined, Arun. Last time we covered a story, maybe not the last, but the last I remember from the NEJM was Masayo Takahashi's story about the autologous iPS-derived retinal cells. And you know when that was, Arun? March 2020 or 2017 is when that was published. So we're talking approaching 8 years ago. And that's what I mean in terms of like the agonizing progress. But again, necessary. In that case, there was one patient, right? And I'm looking at the abstract here. A year after surgery, transplant remained intact. Best corrected visual acuity had not improved, but also had not worsened. And cystoid macular edema was present. So that's probably not good. And ultimately, we all know how that ended up. The autologous approach was not abandoned, but I think was put aside for this allogeneic. And here I'm impressed because we've moved past those, I would say, tepid results in that story, to show some real efficacy and to show the safety, I think, is the real key thing. And yeah, you mentioned the mild and moderate, but the key being there zero serious adverse events, right? So I think reaching the endpoints on safety, A plus. I'm a little bit more, I'd say, conservative on whether they reach the efficacy endpoints. I think they're celebrating this as a win, and I would say yes, it is a win. But again, the progress is agonizingly slow, and And my hope is that you have this kind of shotgun approach where enough of these things, and it's true, enough of these things are going on in parallel that the results will, dribble out and then really be, I like no exaggeration, on a weekly basis. I don't know, daily maybe even, where we're getting results from these trials as the volume increases, and like I said, that the timing, the turnaround on them finally matures, and we can see the results. So yeah, I think exciting progress. I think that we've got such a long way to go, and it's going to be looking at I don't know that we're accelerating, but I think it's going to be, decades before we're looking at the full maturation and dissemination of these therapies widely. But it's very exciting to see that we got cells that we're no longer measuring by whether the cells remain there. That used to be the metric. Hey, the cells are still there. Now we're expecting to see some improvement, and that's what we're getting.
Yeah, I think it's an exciting time for the field, and I'm going to be maybe a little bit more optimistic. I'm going to say by the end of the decade, there's going to be an approved iPSC derived cell therapy out there, whether the indication is for the eye or for the heart or for the brain. I think this is going to happen just because of the breadth of the scale and the scope of the therapies that are emerging over the last few years. I mean, sure, I think the efficacy part of it has to be fleshed out when it comes to this particular therapy, but the safety is really promising, and that's all you can really get at for an N of four eyes, very small, single arm study like they did here. We didn't talk about the economics of this at all, but inevitably that's something that does have to be discussed anytime you're talking about cell therapies. I mean, one way to somewhat alleviate the concerns with the economics is certainly using allogeneic as opposed to autologous, more of like an off the shelf approach. But regardless, a really exciting time for the field.
Yes, and to be clear, I agree with you that we're going to have approved therapies, the economics notwithstanding. They're going to be something approved by the FDA. I agree with you on that timeline. I'm talking specifically about the eye. And I think it's notable because the eye was at once this symbol of like, oh, this is the eye, immune privilege. This is going to be the front line of cell-based therapy. And I think that optimism has kind of, and not in a bad way, but other things have kind of caught up and maybe advanced beyond where we are in the eye. But also, like, metaphorically speaking, it has that Jesus element, right? I was blind and then I could see. So I love the idea of the eye, and the eye was targeted much like the heart and spinal cord, because there's such a vivid uh, uh, you know uh, illustration of the power and the hope. So that's what I want to say. I want to put that all in context. Like, it's agonizing, it's all positive, but for me, because it's such a hopeful symbol, it's I wish it were happening at a different scale, and I think it's going to take some time. But I agree with you that therapies are on the way. People, get ready, get some cells in you. Spending a little bit too much time on this story, but I think it deserved it. A clinical story like this, we got to talk more about those, and we'll have plenty of opportunity as time moves on. Moving on to another story here. I just want to start this one by saying that this was like the atlases have been coming. We've been covering it as a skin atlas. We covered on a previous Roundup in a previous episode. A lot of atlases have been coming out at a steady stream, but this week was like specifically this one issue of Nature that advanced online, published on the 20th of November, went Rambo on the atlases. I mean, just to name a few, and there's other journals, but in this one Nature journal, there's one from Aviva Rega, led by her group, about just like, you want to identify cells, period, across the board, which is comprehensive. Check it out. Check out this whole Nature issue. One from organoids from Barbara Trutling, like all the organoids consolidated into one, and the disease states and others. And then Sarah Teichmann went insane. I've never seen this, Arun. Two papers in Nature that were atlas, one a spatial human thymus atlas in Nature, and also that she led, also this human embryonic skeletal atlas in Nature. And then the story I'm going to cover was in Nature Medicine. When you get a stem cell story in Nature Medicine, it's kind of a big deal, because nature medicine is like exclusively focused on clinical things that are not just like clinically relevant, but clinically now, like in practice. So this last one, near to my heart, loose pun there, is about the the vasculature. When I love my vessels, and this is this complete, comprehensive atlas of the human vascular system, which we know, I've said it a million times, angiochrine, boss, and talking about the heterogeneity. It's not just plumbing. Shaheen, my dear mentor, really impressed this upon me. I carry it with me. It's not just about blood flow gas exchange, right? And in the lymphatics, removing interstitial fluid. There's like the architecture. There's all the other function, the coagulation, immune regulation, tissue repair, and like, yeah, even the shape of these things. You have fenestrated, sinusoidal, but like the molecular characterization, while we know about this whole angiocrine, there's all these different organotypic endothelial vascular cell beds, while we know they exist, an objective characterization of them all side by side has not come to the fore yet. And Shaheen, where were you, partner? I would have expected you to be coming out with this, long time ago. I see you be collecting endothelium off of roaches in your office. You're insane. But Sarah beat you to it. And here, and Avera, I think you would be very pleased, and I'm sure you're pouring through the data set right now, very pleased with this effort. There was, not to put aside just it's a general resource, but they illustrated the utility of it. And by the way, not just endothelium, but also mural cells, and the interaction between the two. But they found, of course, that there are these functional differences across the vascular beds, and zeroed in on that in 19 human organs and tissues. So not everything, but a lot. They had 67,000 cells from 62 donors, and they identified 42 distinct vascular cell states, which were like, the across many axes. The interesting ones to me were like the caliber, looking across the endothelial or the arterial caliber from large to small caliber vessels and capillaries, looking at the molecular profile and differences there. Looking at specific organotypic populations, like in the spleen, blood-brain barrier, which are, the classic. And then again, as I alluded to, looking at the endothelial mural crosstalk, showing that there's in specific organs, there's different varieties that include Notch and/or Wnt, retinoic acid, prostaglandin, cell adhesion signaling. They looked then deeper at transcription factor networks, showing that in different organs, there's of course a different purview of targets and master regulators. And then here, highlighting the clinical value here for nature medicine. I'm sure this was pleasing to the editors and reviewers. They inferred the kind of mechanistic impact of drugs on these specific vascular beds, which is critical because, like, I feel like we're completely in space. I mean, Arun, you appreciate that, the idea of off-target toxicity, specifically in the heart of like chemo, for example. And we talk about organ toxicity for different, don't drink grapefruit, all that stuff. But like the effect of drugs on specific vascular beds, I don't think anyone talks about it at all. So I love that they provided this resource, showed how it was useful, and then specifically applied it here in a way that I think is going to be useful in the near term for clinical assessment of off-target drug toxicity. So, Arun, what do you think of this? A feast of atlases, but this one in particular.
Don't drink grape juice, grapefruit. Can you
You
explain
haven't
that?
heard about that? There's like you can't drink grapefruit when you're on this drug because then it has a specific toxicity in the liver or something. You haven't heard about that? I thought you were a scientist,
I,
Arun.
I thought I was, but apparently not at your level of science. You have to send me
I might have made that up. I'm gonna look that off. I'm gonna look that up in the sidebar. I will be right back.
You got to send me the primary literature for that one. Yeah, but hey, this is a really cool study in Nature Medicine. I know you love this one. I mean, this is what you're all about, the vasculature, angiocrine, Delon angiocrine. I love saying that. But hey, I think Shaheen is going to really love this because one thing that he loves to get to the bottom of is the signature of that brain microvascular endothelial cell, right? That comprises the blood-brain barrier. And I know Clive Svensson here at the Regenerative Medicine Institute at Cedars and Shaheen have gotten into a bit of an argument about the composition and the signature associated with those cells. And that's kind of, we can talk about that offline. There's a whole backstory there. I love you guys. I love you, Clive, by the way. I don't want to get in trouble here. But hey, one of the things like you alluded to that was described in this atlas was the signature for those microvascular cells that are comprising the blood-brain barrier, and those have been notoriously hard to derive from pluripotent stem cells. So perhaps this is going to, help answer that question of how do you actually make those cells, right? Yeah, this is amazing stuff from the Teichmann lab. Incredible productivity. I also love how openly available these data sets are for accessing by the scientific community. You can just go to github.com/tychlab/vascular atlas, or you can even check out the drug to cell atlas as well. I'm sure Paul Burridge is going to love to take a look at this. Speaking of, toxicity, vascular toxicity. In fact, one of his, I guess, his probably biggest study as a postdoc in Joe Wu's lab was also in Nature Medicine. It was looking at doxorubicin toxicity in iPS-derived cardiomyocytes. So there is a precedent for this area of work in Nature Medicine, but it takes a lot of work to get in a caliber of journal like this. I think there's a lot of stuff to unpack here, but in general, just a great resource for the vascular biology community.
Yes, for the vascular community, for this skeletal muscle, thymus, she's doing. They be atlasing over there in that lab. And it wasn't in that lab, but this neuroorganoid one from Trutland. I know I'm talking about other stories that we didn't cover, but I invite you guys to take a look because I think it illustrates the utility of this whole era of like post-publication science, right? Aggregating all these data sets can be a paper in itself because there's so much more discovery. And I think it can inspire all of us to there is an atlas that's relevant to what you care about, so you can go out there and maybe generate some hypotheses and just a final kernel for you. Arun, grapefruit juice can interact with many drugs, okay, and it blocks an enzyme that helps your body metabolize medications. So there it is. Now take that with you after the podcast. All right, you're welcome.
That was like the broadest conclusion I've ever heard in my life. Thank you for that information. Moving on to something very different, and this is going to lead into our conversation with Mansi Srivastava, who's focused on adult pluripotent stem cells and sort of the evolution of those mechanisms and how those really cool capabilities in the flatworm and other animals emerged. We don't have adult pluripotent stem cells, obviously, in humans, but some lower vertebrates, lower invertebrates have these capabilities. And so the paper I'm going to talk about next is in that realm of the evolution of pluripotency. This is a Nature Communications study coming from Ralph Schausch over there at the Li Ka-shing Faculty of Medicine, University of Hong Kong. Shout out to Hong Kong. We're super excited to go there for ISSCR next year. Can't wait to maybe say hello to these folks and congratulate them for a really cool study here. This is looking at the evolution and the emergence of the SOX and POU transcription factors, and ultimately indicating that the emergence of these critical transcription factors for pluripotency predates the origins of animal stem cells. When we're talking about SOX, we know SOX2, POU, commonly known as the OCT transcription factor. These are two of the hallmark transcription factors thought to be associated with pluripotency. I mean, they're two of the canonical Yamanaka factors, right? SOX2, OCT4. And, one thought is that stem cells, which are so critically regulated by these canonical transcription factors, are a hallmark of animal multicellularity, right? Stem cells, they turn into other cells, they differentiate to other cells. So you would think that they're intrinsic to multicellular animals, right? Kind of just by definition. But apparently, these SOX and POU transcription factors go beyond or before the evolution of multicellularity, and they were previously believed to be only a multicellular animal innovation associated with stemness, of course, but were thought to be absent in unicellular distant relatives of us in the animal kingdom, right? But here they're actually describing that there are unicellular SOX and POU factors, transcription factors. They found them in the choanoflagellates, these unicellular animals that are found in all sorts of water in the ocean and ponds and stuff, as well as the Philisterians, another unicellular subset of organisms. Both of these, choanoflagellates and Philisterians have SOX proteins, and they get this, they actually have a DNA binding specificity similar to the well-characterized mammalian SOX2. The choanoflagellate but not Philisterian SOX can get this actually replace standard SOX2 to reprogram mouse somatic cells into mouse iPSCs.
Wow.
So that's literally what they did. They took the SOX2 or the whatever evolutionary analog from the anaflagellate, expressed it in mouse somatic cells, and we're still able to reprogram those mouse somatic cells into mouse iPSCs. That's amazing. That's an amazing evolutionary study. And it happened mechanistically because those choanoflagellate SOX proteins interacted with the mouse OCT4. So even though these animals are millions and I don't know, a billion years apart on the evolutionary tree, their canonical transcription factors driving pluripotency can still interact with each other in a capacity to actually generate iPSCs from the choanoflagellate SOX2. This is just an incredible story that just kind of blows my mind. In contrast, the choanoflagellate OCT4 actually has a DNA binding profile that's very distinct and cannot generate iPSCs. So there's a distinct difference between the choanoflagellate POU or OCT protein and the SOX protein. The SOX protein can work to make those mouse iPSCs, but the OCT cannot. And they then ancestrally reconstructed the lineage of the SOX proteins and indicated that iPS formation capacity is pervasive among a bunch of resurrected sequences from back in the day, these very early stage unicellular organisms. And that the loss of SOX2 like properties actually fosters the generation of other submembers of the SOX family. Okay, so it's a reflection on how critical the SOX protein is in generating iPSCs for one, but also for maintenance of the pluripotency network across the animal kingdom for two. And ultimately, I think it's a pretty big conclusion that they're getting at here that the evolution of animal stem cells might have involved the adaptation of a pre existing set of transcription factors like SOX, where pre animal SOX is actually biologically similar to the SOX that's around right now in mice. Right. But the OCT factors, the POU factors, have a totally different evolutionary origin. This is I thought, a mind blowing study. Really kind of the fact that you can generate mouse iPSCs from a SOX factor from a unicellular organism that's a billion years evolutionary distinct from the mouse. The fact that that worked is science fiction stuff to me. I thought this was a really cool study.
Super, that's a twist, man. I hearing you talk about it, I'm looking at the story and it's like raising a lot more questions than answers for me. Not in like cynicism or doubt, just like what? Like as you're saying there, weird, right? Unexpected. And yeah, it makes sense to me the from a teleological standpoint, like yeah, you take what's there, right? Repurpose, sure. That's what nature does. But it's the conservation that's so weird and I can't wrap my head around that the like I guess functionally speaking, I mean clearly it's not doing the same thing, right, in a unicellular organism, but like that the activity can be transposed into a mammalian cell and have the functional equivalent of, the ortholog. It's weird because I'm just like, what's it doing? What's it doing in the organism of origin? Is there a discussion of like what's the functional role in the cell? I mean, I know SOX probably something metabolic, I guess. I don't know, but it's just one of those papers that, yeah, as you say, is so interesting and impactful just because it really is a head scratcher.
Yeah, I think a lot to unpack here. I'm not sure about the role of SOX itself in that coinophagia or whatever unicellular organism, because again, like it's unicellular. It's not a stem cell per se. It can't turn or differentiate into other cell types. It doesn't make other cell types. It only has one. But it's still alive. It's still an animal for sure. It's just the simplest type of animal. And you're right. Maybe the SOX factors or these early pluripotency factors have completely different mechanisms or utilities in those creatures found in the ocean. I don't know. A lot to unpack here. But I love thinking about this stuff. It just really gets the sci-fi fan in me going, and I know you're a big sci-fi fan too.
Oh, absolutely. And yeah, it's important, I think, to always get back into the basics, to get outside of our, cell type or species comfort zone. And here you went deep, partner, literally. The story I have is kind of in the same vein. And I like it because it really takes me back. I'm nostalgic for my graduate school days where there was just a lot of basic, just what are the pathways that are critical for maintaining pluripotency, for example, was a big target. Everyone was trying to make a recipe to keep cells going, something that didn't involve meth, that didn't involve matrigel, maybe defined factors. That was it. That was the race. Right. And along with later induced pluripotent stem cells. But the early, turn of the millennium, soon after their derivation, we were trying to find ways to keep them going. And a lot of that was understanding the pathways that modulated these first specification events. And we assumed that what was going on in culture and ES cells was a mirror of what went on physiologically. And that has not always been the case. I mean, we recognize very early that human embryonic stem cells are, an artifact. They only exist transiently in vivo, and our whole system that we have here is a great big hack. But getting back to this story, we're talking about like lineage specification. The first one being going from a zygote to trophoblasts, inner and outer, right? That polarization, and the second is the differentiation of epiblasts from primitive endoderm, which it's known in mouse is governed by FGF signaling, um, and there's a similar phenomenon, the epiblast to primitive endoderm divergence in human. We know looking at embryos, but like the signaling mechanism that drives that is not very well known. Although a lot of assumption goes to TGF beta signaling. TGF beta signaling is plays a prime role in a lot of early developmental events, in a lot of events, period, right? But TGF beta signaling is known, thanks to yours truly, if I don't say so myself. A little humble pat on the back. And another reason I chose this story is because my first author paper from my graduate thesis, first paper in my life, but it was the first of my thesis, was cited here. So much respect to Kathy Nyakin's group, who are at the Human Embryo and Stem Cell Laboratory. So you know what they do there at the Francis Crick Institute. But thanks for the citation. And again, going back to what I was saying, that's kind of circling back to these old ideas, first findings. Ludovic Vallier, who we just had on the show, also heavily cited here for the same kind of science. But the point being is that we had our finger on TGF beta signaling as a primary modulator of pluripotency in stem cell culture. And here, the question is that the case also in the human embryo, right? The only TGF beta ligand and receptor pair that's expressed in pre-implantation human development is nodal, the receptor, and co-receptor or a ligand, co-ligand, sorry, and GDF3. Um, and in mouse embryos, nodal signaling has been detected in pre-implantation stages, but there are some differences there. But it suggested that nodal signaling is involved in pre-implantation human development. Here, using old school methods again, but also embryo, live human embryos, not human embryo models, which I will come back to because I think it'll be interesting to revisit these questions in the embryo models. And I'm sure those experiments are ongoing. But here, using the real thing, they were able to track like real-time activation of nodal signaling. What they showed is that NANOG, of course, pluripotency marker is a marker of nascent epiblast, right? And the key is that that fate is initiated prior to the activation of nodal signaling. And this is the real key takeaway from this story. It goes against the dogma, this idea that nodal signaling precedes the acquisition of pluripotency and is essential for the maintenance. This kind of unsettled that. It showed that the story also, they showed that the pluripotency factors, NANOG, SOX, OCT, KLF, they were maintained even when you blocked nodal signaling. So not just showing that the identity preceded, but like nodal signaling wasn't even necessary in the context of human embryo. So that's a very, again, a basic study, but a big finding, suggesting that there's pretty significant differences in the signaling requirements that regulate pluripotency, like in real life, in vivo, in a physiological context, versus the what regulates and is essential for pluripotency in ES cell culture. And to me, that is not as big a twist as like what the hell is SOX doing in a unicellular organism. I'll admit I'm going to be scratching my head bald on that one. But in this case, it's like, wait, what? It's kind of a reset. Like we've been going on this assumption for so long about the essential role, like critical role of TGF beta signaling, specifically nodal. And here we go. In embryos, not so much. So I think it's an important reminder and underscores this great big hack. And it'll be interesting, as I alluded to earlier, to come back in human embryo models to these phenomena and see. I mean, I wouldn't be surprised either way. Maybe the human embryo models do it a different way, are more like the stem cells in culture, than they are than the bona fide human embryos. Like that's a question in itself. So, and a way to like quote unquote validate these human embryo models, perhaps. I don't know. A lot of questions again from this one, scratching the other side of my head.
Yeah, a really great basic study here. And like when you first sent this paper over to me and I looked at the figures, I thought it was a study focused on early embryo models because we are so entrenched in that field and it is the hottest subfield in stem cell biology right now. But no, these are true human embryos derived from donations, early phase human embryos. And it's, refreshing, I guess, in some ways to see a study like this. And this is really, of course, what they do over there at Cambridge and at the Crick. They have such a history in this area, as you're very well aware. I mean, one thing to mention here, and they actually mentioned it in their limitations. They use this inhibitor A8301. I believe it's a small molecule, and it can impact both nodal and BMP signaling at high concentration. So some of the conclusions you have to make, at least transcriptomically that they make here, might not be exclusively attributed to inhibition of nodal signaling alone, just because there could be some of these off-target effects. But I agree with you. I think it'd be really nice to revisit some of these mechanisms using the early embryo models and really see how comparable those things are to the real deal, right?
Oh yeah, and then you bring in the whole other dimension of the engineering, right? You can do the knock-in, knock-out. You can do the lineage tracing and reporting. So yeah, it's going to be back to basic. It's really, we say it all the time. Wonderful time to be alive, right? As a scientist, but I think most of the time we're talking about therapeutically and translationally. I think the basic developmental biologists, this is the year or the years that really highlight and are extending the tool set for those basic questions. This being a really great example. So looking forward to the Nyakin group turning their focus on some human embryo models, maybe. But as you said at the Crick, they keep it real, literally. And again, I want to highlight how, as you said, using these human embryo models donated, there still really is an important place for human embryos and fetal tissue, and studies like this really draw attention to that. Moving on, we had Scott back to basics here on the show, started with some heavy translation, and here we are headed into the interview, going back to square one almost, but now we're going in a different direction, kind of evolutionarily. Arun touched on it with the flagellates, unicellular. We're going to go a little bit higher. I don't want to place value on it, but on the evolutionary ladder, at least in terms of cellularity speaking, we're talking about many cells, but cells that exhibit some really tremendous qualities. We're talking about that with Mansi Srivastava in just a minute. But before we get there, I have a quick message from STEMCELL Technologies, who's inviting you to find a pathway into the peripheral nervous system with STEMDiff Neural Crest Differentiation Kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, STEMDiff Neural Crest Differentiation Kit enables high purity generation of neural crest precursors from human pluripotent stem cells. Learn more at www.stemcell.com/NCKit. That's NCKIT. All right, everybody. Today on the show, we have with us a special guest from Harvard University, professor of organismic and evolutionary biology, also curator of invertebrate zoology at the Museum of Comparative Zoology. That's one of the oldest research museums in the country at Harvard University. We have with us Dr. Mansi Srivastava. The Srivastava Lab seeks to identify cellular and genetic mechanisms for regeneration by comparing mechanisms across distantly related animals, and I mean distantly. The main model in the lab, the acellular worm, Hofstenia miamia, aka the three-banded panther worm, belongs to a lineage that is distantly related to traditional regenerative model invertebrates such as planarians and Hydra. Mansi, thank you so much for joining us on the show today.
My pleasure to be here.
Yeah, welcome to the show. And as Dalon knows, we love discussing unique model systems here on the podcast. I think it also gives us the opportunity to maybe nerd out a little bit because we're both big science fiction fans. And in sci-fi, there's that common theme of superhuman regeneration. Wolverine from X-Men comes to mind. And indeed, you work with the unique model system in Hofstenia miamia, the panther worm, which also has this superhuman or super worm regenerative capacity thanks to its adult pluripotent stem cells. And before we start nerding out about this amazing worm, can you just start things off by giving our listeners an overview of what your lab is working on, kind of in your own words?
Yeah, sure. So as you guys already brought up, these worms are super regenerators. You can cut them in 2 pieces, 3, 5, more, and each piece will go on to make a whole new animal, which means it's making a whole new brain from scratch that's going to become fully functioning and allow the worm to be a whole organism at the very end. So in the context of this property of whole body regeneration, we ask many questions, which fall broadly in 3 processes. First, you imagine when you cut the worm, immediately the organism figures out something has happened and a whole bunch of genetic processes are launched. So one of the objectives in the lab is to use a gene regulatory network framework to figure out how the worm figures out it's injured, and then it launches all of the cellular and molecular pathways needed for this whole body regeneration process. Now, another major line of inquiry in the lab pertains to the source of new tissue, right? When you cut the worm and it's going to make a new brain, all of those cells have to come from somewhere. One option would have been that you take your existing brain cells and somehow make them it's lost a brain, so it can't take its existing cells, but maybe there's a process of transdifferentiation that could have occurred. But instead, what this worm does is it utilizes this large population of adult pluripotent stem cells. Sometimes we abbreviate those as APSCs, that are then mobilized. They figure out stuff has gone wrong, and then they divide and divide and divide and make all of the cells that have gone missing. So we try to understand what endows these cells with these really awesome capacities. And a third aspect we are trying to understand is, how do these cells know every time exactly which cells to make? Because if I chop the head off, these cells are going to make a brain. If I chop the posterior off, they're going to make a new tail. And so there's a lot of patterning information in place that every single time, without making mistakes, this worm knows what's where. And so we're also trying to figure out how these systems are maintained and then regulated upon wounding. So I would say, in relation to regeneration, those are the three major aspects my lab is studying.
Yeah, I mean, you talk about those three things really in the context of this panther worm, but I mean, there's an argument to be made that those are like the three things in developmental biology, period. And on the show, we talk a lot about pluripotent stem cells. We're going to talk a bit about those, although different kind of pluripotent cells, those APSCs. But on this show, we talk about pluripotent cells, and most of the times human, right? These days, and the exciting applications of those cells, mostly therapeutic nowadays, which is great, very exciting, been a long time coming, and it's a great time to be alive as a scientist. But we're all, I think, arguably developmental biologists at heart, and in practice, really, no matter what we're doing, what we're working with is this clay that is a tool for furthering insight into developmental biology across systems. And that's been underscored, I think, recently by the advance of these human embryo models, having drawn a lot of renewed attention to the power of these model systems, just fundamentally why we do the science, to reveal the mysteries of our origins, from day one, Aristotle looking at the egg. But for the entire history of developmental biology, while nowadays we're using these human-ish model systems, animals have been the main model, right? I looked this up. Around 20 million animal subjects are used globally every year in biomedical research. Of course, the vast majority are rodents, although less and less each year. That's interesting. Both this diversification of other models, also the cell-based. But particularly, I've been really delighted in recent years of these more exotic model systems that have emerged, precisely because they're so different from human development or physiology. My favorite, as like a reproductive biologist, is this naked mole rat, which is both hideous and beautiful. Hideous looking, although cosmetically, to each his own. But such a beautiful biology there, from the eusocial organization that's akin to like bees or ants, to the cancer immunity, to the lifespan tenfold your other rodents. Like, all those things are so far out, like Arun was talking about, like comic book stuff. So I'm asking you, as someone who's developed this exotic model of your own, tell us what drove that interest in the panther worm specifically as a model, and perhaps what was like the most challenging aspect of establishing or working with this system?
Yeah, well, you said a lot of things there that are really important, and to think about a bit deeply. We think of these species as exotic, right? Because we are doing science in a framework where we have narrowed down to just a handful of model systems. But historically, these systems were not exotic, they were the norm, right? So if you look back at even just a little over 100 years ago, Thomas Hunt Morgan, who is now credited as the founder of genetics, was spending his life traveling to the Marine Biological Laboratory every summer. And what was he doing there? He was chopping up worms, right? And so this is a place where developmental biologists would travel to every summer, because lots and lots of marine species were spawning and making egg and sperm, so you could fertilize in the lab and study these organisms. And it used to be 120 years ago that people studied many, many, many more species. And so in that context, while the species I study seems exotic, in this larger context of developmental biology, as you pointed out, it's not that exotic. But there are some new standards that these exotic species have to meet, right? Because over time, developmental biology narrowed down on a handful of model systems because we wanted to delve deeper into mechanistic depth, right? We needed to understand how genes work, how cells work, and so on and so forth. And it makes sense that it's hard to do that in a lot of different species at the same time. But now what's happened over the years is that the standards for making an inference in developmental biology went way up, right? We need to do certain types of very rigorous experiments. And so a challenge now in bringing these currently understudied species back to the lab is to make sure that we're able to apply that same level of rigor that we've been able to apply to Drosophila, which is fruit flies, or C. elegans, which are roundworms, and other models for developmental biology. So I think getting back to your main question of, what were the challenges? So first was figuring out which species to study. So my motivation for branching beyond the organisms that were already well studied in the lab was coming from an evolutionary biology perspective. To me, fundamentally, what I find exciting about biology is trying to understand where things come from, right? So it's cool to me to figure out how things work. But another question I like to ask is, does it have to only work that way, or are there other ways this could have worked? And evolution has done these natural experiments, right? And so one of the questions I was driven to address was, say we study regeneration in one species or development in one species. Is that the rule or is that an exception? To be able to answer that question, you have to have other species that you can compare those processes to, right? And then once you agree that we need to study the process in multiple species, you might want to think about what is the right phylum or evolutionary distance that you would want to branch out to as you expand your repertoire of organisms. So actually, I tried working on lots and lots of different other species during the first year of my postdoc. So my postdoc was in a lab that studies planarian worms, which many folks might be familiar with already. These are the googly-eyed worms that are freshwater. You can walk into pretty much any stream anywhere in the world, turn a rock over, and you'll find one of them. And they're also master regenerators. So what I was hoping for was finding species where we could study genetic pathways for regeneration, but the species would be distantly related from planarians so that we can make some statements going deep into time. And so the biggest challenge was you bring a few species to the lab, and first, some of them don't live at all because you can't figure out what to feed them or the right water or temperature conditions. Second, they might live, but the kinds of genetic tools that you'd like to apply to them don't work. So you can cut them and watch them, but you can't really get down to which genes might be operating and controlling regeneration. So because of these failure points, various species that I tried to work with did not end up progressing. And Hofstenia miamia, which is an acial species, just was the one that we got very lucky with. So it was, we went after Hofstenia because it belongs to this phylum called the Xenacoelomorpha, which has a cool property. One, they're wormy. So they're easy to compare to planarian worms at an anatomical level, right? You can line up two worms next to each other, you know where the heads are, the tails are, and the backs and the bellies, and then you can chop them up in the same ways and ask questions. But the other cool property was, despite being wormy, they were very distantly related to planarian worms. The last common ancestor that gave rise to planarians and acials and to humans existed 550 million years ago. So if we find things to be shared between planarians and acials, we can hypothesize that they were present in that last common ancestor, and then we can test that hypothesis. So focusing on this phylum made a lot of sense, but there were no perfect species that were currently being studied in the lab from that phylum. So we actually went to the field we had talked to taxonomists who study ACLs all over the world about which ACL could we bring to the lab, where could we collect them. And there are papers published that had found that Hofstenia miamia lives among mangrove environments. It eats crustaceans. So we thought, oh, maybe we could bring them to the lab and feed them brine shrimp, just as one would feed zebrafish. And in a study from 2007, there was an exact latitude and longitude location for where these taxonomists had collected this species, which happened to be a marine pond in Bermuda. So we, booked our flights to go to Bermuda. I sought the help of the Bermuda Museum of Natural History, so that they could point us to which part of the pond matches that latitude and longitude location. And we found the worms there. So the challenges that we overcame were first, I was allowed to collect 120 worms, which is exactly the number we brought back. You have to go through all of the permitting processes to bring different species across international borders. I got super lucky in that I was able to culture them in the lab. We tried feeding them liver, which is what planarian worms eat, but they hated that. So we realized they needed to eat live things. And that makes sense because they are a predatory species. So they like to go on a little hunt after living things. And that worked well. And then as a big surprise, they started producing egg and sperm and making babies. So then I had super tiny, tiny worms, which was exciting, but they were too small to actually eat the brine shrimp. So then we had to figure out, well, what are these guys going to eat? And I ended up calling my undergraduate advisor, who seems to just know all kinds of things, and he recommended that I try feeding them rotifers, because rotifers are a whole other marine invertebrate phylum that is smaller. Organisms are smaller than the baby worms that Hofstenia were producing, and so that's how we got the worms cultured in the lab. They go through their life cycle. So egg to egg only takes about 4 to 6 weeks. And then we tried various techniques like sequencing their genome, sequencing their transcriptome, developing methods for looking at gene expression, developing methods for perturbing gene function using RNAi, and finally also making transgenic worms. And all of those worked. And there's definitely some element of luck there, because when you pick a random species out of the ocean, you don't know which techniques will actually work in it. So combination of luck and perseverance paid off for us.
What an amazing story and backstory there. I mean, that's there's a lot there. I think, anytime you want to be a pioneer, like what you're doing here, you have to take this leap of faith. It's so hard to do these things from scratch, which is really what you're doing in a lot of ways. You got to figure out how these things eat, what they eat, how to make them happy and grow, how to figure out the reproduction, the genetics, the transgenics. I mean, this is years and years of work, luck, and a lot of hard work. That's definitely for sure. And there's, I suppose, more than one reason to go to Bermuda. I suppose you can go sit on the beach, or you can collect some worms.
That's right. I suspect that some proportion of the trainees in my lab, when they joined, were hoping we might end up in Bermuda sometime. Unfortunately for them, we haven't had to go back. So I had originally collected the worms in April of 2010 and haven't returned.
Never say never, I suppose.
That's true. Well,
yeah.
Hopefully the worms are going to make it.
Yeah, fingers crossed. Let's dive a little deeper into the science. You had this Cell paper a few years ago that really took the stem cell field by storm, where you described the embryonic origins of the adult PSCs and H. miamia. I mean, a really cool concept, like what we're talking about. But in particular, those of us in this field who are so entrenched in IPS biology, induced pluripotent stem cells, where we're just so hardwired to believe that true pluripotency only exists in this embryonic state during mammalian development, for example, which is what most of us work on, I guess, right? On the other hand, in the panther worm, there's these adult pluripotent stem cells called neoblasts. And then you did a bunch of lineage tracing, single cell, showing that the adult PSCs in the worm are derived from one lineage, not from multiple tissue-specific lineages during development. So take us through that paper, the Cell paper from a couple years ago, and maybe
also tell us a little bit more about the utility of the single-cell developmental atlas that you actually made for the worm.
Yeah, sure. So actually, to start talking about that, I'll go back to something that Daylon mentioned. You were stressing this deep connection between regeneration and developmental biology, right? That most of us are really studying development. And so one of the things that became clear to me early on, once we started culturing Hofstenia in the lab, was that it's a really great system where we can not only study regeneration, but also study development, because they're constantly making these embryos, which are accessible. We can pick them up at any stage, starting from the zygote, and ask questions. And one of the questions that I became drawn to, particularly guided by the interest of one of my first graduate students, Julian Kimura, who was deeply trained in embryology, was about how do these stem cells get made in the first place. So just to give you a quick recap about these adult pluripotent stem cells that we're talking about, in a species like Hofstenia, these are the only dividing or proliferative cells in the system, and therefore they are the only source of all of the new tissue. And in some species, such as planarians, it's been shown that even a single one of these cells is truly pluripotent. You can take a single cell, put it in a worm that doesn't have any of its own stem cells, and this one cell will produce all germ layers and all cell types, showing that its pluripotency. In other systems, we know definitively that they're collectively pluripotent, and we're still awaiting evidence for individual cells being pluripotent. But regardless, what stands out here, hopefully, is that Hofstenia is not the only worm on our planet to have this type of stem population. This is a population we see across animal evolution, ranging from species like sponges or cnidarians like Hydractinia, which are hydroids, and jellyfish. We also see stem cells like this, as I already mentioned, in aseals, planarians, but also in invertebrates that are much more closely related to vertebrates, which is us, which is a phylum called the tunicates. So now this adult pluripotent stem cell population is widespread across animal evolution, which is crazy, right? Because as you mentioned, in a human context, we think of true pluripotency only existing in very early embryonic stages. So it's really exciting to ask this question of where does this pluripotency in adult cells come from? And although these adult pluripotent stem cells, which are also called neoblasts in aseals and planarians, all referred to as eye cells and cnidarians, they've been studied in the context of adult biology in all of these exotic species. In no species did we have an answer to how they get made. And the sort of null hypotheses we had going into this work were the following. One, maybe these cells are quite like human stem cells. So remember, you and I don't have pluripotent stem cells, but we have plenty of stem cells in our bodies, right? We have intestinal stem cells. We have stem cells in our hair follicles. The problem is that intestine stem cells are not going to turn in and make you hair follicle stem cells or vice versa. So they're lineage committed or lineage restricted, as opposed to being pluripotent. So Arun, you mentioned the single cell atlas study we did in Hofstenia. It gave us a view or a window into the molecular properties of all of the cells in Hofstenia at single cell resolution. And it revealed to us that much like has been reported in planarian adult pluripotent stem cells, these thousands of cells in the body are actually heterogeneous. Many of them already look like they might be on their way to making muscle. Many of them look like they're on their way to making neurons. Many on their way to making skin, so on and so forth. So now it could be that the way this population gets set up in Hofstenia is exactly how stem cells in my body got set up. That when I was an embryo, the same lineage of cells that made my muscle, the functional muscle cells that make me move my arm, also that lineage gave rise to muscle satellite cells. And the same lineage of endodermal cells that made my intestinal lining also gave rise to intestine stem cells, right? So you could imagine in Hofstenia also there would be multiple origins of these cells. That in the Hofstenia embryo, there's some lineage of cells that's going to make ectoderm, and those could make the more skin-like, skin-aligned stem cells. And the lineage in the Hofstenia embryo that would normally make muscle will make more muscle-oriented stem cells, so on and so forth. So you could imagine a scenario of heterogeneous origins of the heterogeneous stem cell population, right? Because the stem cells in our bodies also have heterogeneous origins. And the alternative could be that maybe there's one single origin. I would say that potentially my favored expectation was that there would be just multiple origins of these stem cells. So now let's come to why Hofstenia is the first species where we were able to pose and address this question. And that's because many of these other exotic regeneration models, they're great for studying adult regeneration biology, but they're less good for studying embryology. Hofstenia is a great exception where it's a marine species. It gives rise to these free living embryos that are available to us from the single cell or zygote stage. And another cool property of these cells is as the zygote starts cleaving or dividing, it does so in a very stereotyped fashion, such that embryo to embryo, the cleavages will occur at a very specific angle, at a very specific time, and the spindles will be located at very specific spacings, controlling the symmetry or asymmetry of the cleavage. So when that happens in an embryo, maybe folks in the audience might be familiar with a C. elegans embryo, which is also a stereotype cleaving embryo. And famously, Nobel Prizes were awarded for people figuring out exactly how these early embryonic cells give rise to very specific adult tissues in the worm, right? So we wondered if in Hofstenia also, because the cleavage program was so stereotyped, whether we could ask what the cellular sources of all of the adult tissues would be. So Julian Kimura, the student who I mentioned, took a systematic approach. And he also got really lucky because one of the postdocs in my lab, Lorenzo Ricci, had generated a transgenic line where every single cell of the embryo is glowing green because of the expression of this photoconvertible green fluorescent protein called CATA. And when you shine light at 405 nanometer wavelength onto this protein, it converts to red fluorescence. So with Lorenzo's transgenic line in hand, what Julian started to do was systematically convert all individually, cell by cell, each one of the cells in the early stage Hofstenia embryo to ask whether they give rise to specific tissues in the adult animal. So for example, if you have the 8-cell stage embryo and you photoconvert the 2 cells at the very top of the embryo, in every single experiment, those 2 cells will give rise to the entire nervous system of the animal. So now doing this for all of the other cells as well, Julian discovered that there was this one pair of cells at the 16-cell stage. Now, when you photoconvert, they always labeled this large population of cells in the middle of the animal, whose distribution was very, very reminiscent of these adult pluripotent stem cells. So this was very exciting because that it led to the hypothesis that these 2 cells at the 16-cell stage, which we refer to as 3 little a and 3 little b, given the nomenclature we use, could be the source of stem cells. But that is a very bold hypothesis, and Julian needed to do lots of experiments to prove that. So one thing he did was he tested whether the progeny of these 2 cells meet the functional definitions of neoplasts. or the adult pluripotent stem cells. So as I had mentioned before, the adult pluripotent stem cells are the only source of new tissue. That's a functional definition, a functional property of these cells. So when we cut the animal and look at the new tissue, if those all the new cells are coming from these two little cells, which now in this experiment we have labeled with red color, then all of the new cells should be labeled with red color. And that's exactly what Julian saw. Now, he also needed to rule out the possibility that other embryonic cells could also be contributing to the stem cell population. And we found that none of the other cells contribute. So it really looked like just these two cells. None of the other cells were producing the stem cells. And then he was able to assess the molecular properties of these cells as well, right? Because using this single cell atlas data set, we could define stem cells at the level of expression of hundreds of genes. And now applying a similar single cell sequencing assay onto the progeny of these three little a, three little b cells, Julian could show that they truly were giving rise to cells that not only look like neoblasts in distribution, not only behave like neoblasts in their functional properties, they also matched the molecular expression profiles of the adult pluripotent stem cell population.
Yeah, I mean, this is, I think, I mean, as you're describing, I'm recognizing, as you kind of alluded to, that this is an old approach with like really newfangled tools, but it's one of the oldest ways we've looked at anything, as you alluded to there with the nematodes, just watching these cells proliferate. In this case, though, in a completely new paradigm, right? And you propose there's like these two, probably more, but two major hypotheses of like the lineage restriction as it is in mammals, or this, truly unique adult pluripotent cell. It's established, as you said, three little a, three little b. And like the mechanism is fascinating, and the lineage tracing, the biology, and the novelty of it. But I think the reason why your story made such a big splash and was a major hit at the ISSCR this past summer is because it's attracted a lot of attention, the work for the clinical implication there. Although you're identifying it as really distinct from this other paradigm of the lineage restriction that's more familiar to us. We have, talked about this kind of idea in the past. Just a couple episodes ago, we covered this paper from Hong-Ki Deng's group describing the de-differentiation of adult cells to these limb bud-like progenitors, which is a really cool story following up his like chemically induced pluripotency. And just a couple episodes before that, we covered this paper in Nature Biotech from Lee Rubin's lab that you were actually on, describing the de-differentiation of myoblasts and the stem cells capable of muscle regeneration. So in this evolving landscape of adult cell plasticity as it relates to regeneration, I think this the notion that the panther worm is truly unique and distinct, exotic, maybe whatever you want to call it, but the notion that it may have some clinical application or implication, or may kind of be applied or deployed, the biology there in the context of human therapy. Is there anything to that? Like, do you see a clinical endpoint for not panther worm cells, obviously, but for the concept you're kind of unraveling here?
Yeah, so I would say, I self-identify as, my science is driven from a basic science perspective. And the way I see it relating to regenerative medicine would be in the same way that in the past 20, 30 years, studies in fruit flies and nematodes have informed regenerative medicine. For example, we wouldn't know how transcription works in human cells if people weren't studying fly embryos, right? Or we wouldn't know how cell asymmetry is established if people hadn't first discovered that in C. elegans embryos, right? So I think so you're right, in Hofstenia, the stem cell population is being established as a single source versus maybe in our bodies, we're getting multiple sources of these lineage restricted cells. What I think the work now enables us to do is to ask a slightly different question, which relates more closely to human regenerative medicine. as you pointed out, that there's this broad idea out there that naturally, or if you look at like native pluripotency, human or mammalian embryonic cells are good at this in the embryonic state. And we can then take our fibroblasts and other cells and put some factors in them and induce them into pluripotent states. But we know that there's cells have memory, right? They still retain marks that are that prevent them from being, truly fully pluripotent in the long term. So one of the questions I've now started to think a lot about is, how is pluripotency being regulated in a species like Hofstenia? Right? Is it that there, just like a mammalian embryo, there is pluripotency in the early cells, and then it's progressively lost? But then this lineage that the 3 little a, 3 little b cells give rise to, they make a new type of pluripotency. It's a whole different new setup. Like, is it special, that something that mammalians could never do? Or alternatively, is it that they're just the few cells that retain embryonic states and somehow keep them going? Because I think that would be a really fascinating outcome, because the key difference between these species is, actually, let me tell you about a quick story from my student Amber Rock, who also came to my lab as a wonderfully trained, curious embryologist, who looked at the Hofstenia embryo and said, hey, it's kind of crazy that they have, every single embryo is going to divide in this very particular way. I want to know what happens if I pull these cells apart. Now, this is a classical developmental biology experiment, right? Where you assess our definitions of whether cells have their fates have been specified, or whether they're determined, or whether they're still pluripotent, come from these simple assays of pulling cells apart and seeing what they're able to do. In fact, this is how embryonic stem cells were made. Somebody said, hey, I'm going to take these cells out of the embryo and see what they can do on their own, right? So Amber did these experiments. She's a very skilled experimentalist. She can dissect the cells without damaging them, and we get very high survival rates. And what she has shown is that even after asymmetric cleavages, these cells are retaining a very high level of plasticity. In fact, one of them is actually even totipotent, which is basically unheard of across the animal tree of life, that an embryo that has undergone an asymmetric cleavage then can still have totipotency from one of these cells. So given that from Amber's work, we know there's a very high level of plasticity in these embryonic cells. And we know that the adult will end up with a large number of cells that are pluripotent. We want to ask the question of how is this pluripotency transmitted to the adult, if at all? Or is pluripotency in the adult cells established as a new thing? Because in a mouse embryo, or when we were embryos, our early cells were pluripotent, and then we lost it, and it was gone forever until the Yamanaka factors came to be. So I think knowing whether their embryonic pluripotency truly cannot be maintained by any animal cell into adulthood, or whether it can be, and figuring out what are the molecular or genetic processes that enable retaining those properties in adult cells, I think will be really informative. So it's not, I think, directly relevant, but it is relevant in the same basic science way through which Drosophila and C. elegans have contributed to human medicine.
Yeah, I think in addition to the science that you're doing, diving deep into the mechanisms of how these pluripotent stem cells work in the panther worm, you also very obviously have a gift for communicating this science. Science communication is something we take very seriously, obviously, on this podcast. And a big element of your lab is actually scientific outreach, telling folks like us and folks in the scientific community and the non-scientific community about these worms and the power of these unique model systems. I mean, in fact, there's an outreach tab right on your lab website. And one of the cool things that you've done as part of this outreach effort is to develop a real science workshop for middle school teachers as part of an NSF career grant. And of course, that's restricted to a grant. You're also doing these initiatives outside of the grant as well. But in this example, you collaborated with the Harvard Museum of Natural History to train middle school teachers and actually train them how to use these modern molecular biology methods for the study of evolution. So tell us more about these really awesome initiatives and science outreach and this initiative and others like it, other efforts that are going on in your lab.
Yeah. So, in thinking about outreach, which I think, I think it is a huge part of our responsibility as scientists to communicate our science to the world and to get people excited about what we do, and also by doing that, encourage the next generation of scientists. And so here I will have to, for this particular program you're talking about, I have to give a credit to my wife, who herself is an educator, and it's through conversations with her that I've learned that a train the trainer model can be a particularly effective way to reach students. That so the way we approach the question, the challenge we were trying to address in designing this outreach program is that people often see evolutionary biology as this armchair science, right? Darwin sat in his chair and then he theorized. And in fact, it's not just so the work I do, I study regeneration and stem cell biology from an evolutionary perspective, right? So I identify in part as an evolutionary biologist, and I think it's really important for not just young students in school systems, but also folks who don't normally embrace evolutionary biology in their day-to-day practice, to see the value in it. So we designed a workshop that allows, we brought in middle school teachers for 3 days. We allowed them to pick anything at a grocery store. Could be spinach, mushrooms, shrimp, beef, what have you. And we came up with a protocol for extracting DNA from basically anything at a grocery store. And so folks come in, they pick whatever they want from the grocery store, they extract DNA from it. Then they use PCR primers to amplify 18S. And we take their amplicons, they run the gel. We took their amplicons and sent them off for sequencing on the second night, got the sequence data back on the morning of the third day of the workshop. And then they were able to do phylogenetic analyses using all of the data that group of teachers generated. And some really cool surprises emerge. You realize that all of the living things in your grocery store are related to each other. And then the second big surprise people get is that mushrooms are more closely related to animal food than they are to plants, right? Because turns out mushrooms are fungi, and fungi are related to animals in a clade called the Opistheconta, and spinach belongs to plants, which are much more distantly related. So it giving this sort of very real feel for how evolutionary biology underlies everything around us, and the fact that you can do it with your own hands, I think gives people a very different way of perspective and level of engagement with that kind of thinking. And the teachers were super excited. We actually then were able to bring our equipment to their classrooms and run bits and pieces of it, whichever worked with their classroom schedules, to get the students excited. And the students, actually, what they reported enjoying even more, everybody loves running gels, everybody loves using pipettes, but even more than that, what they appreciated was getting to meet scientists, which were students from my laboratory and other students in my graduate program. And they're just excited by the fact that scientists are willing to take the time to come talk to them. So that to me was this really powerful reminder of how much power we have as scientists, and how just by taking a little bit of time, we can have a big impact on the lives and thinking of young folk.
Yeah, that's so great. I mean, and that's part of the reason why we are so, dedicated, however dedicated we are to communication, is that those are the elements, right, of of creating the next generation of scientists is one, they need to see the scientists, they need to see themselves, see the possibility of themselves being a scientist, but also that just the demystification of it. Do an assay and say, okay, that's how I did science. I mean, you have no idea how many clinical fellows who are MDs, they've done the most complex surgery in the world. They come to my lab to do a little research stint, 6 months of research. They do 1 day of cell culture. They run one gel, and they're like, I'm doing science. And they start geeking out about it because, it's the stuff we take for granted. But it really is the first step in creating a scientist. They got to do the work, right? And I think that your not only your outreach, but also your example as a scientist is a fascinating illustration of a pathway, how you come to being a scientist here, in your case, at the highest level, right? I mean, just looking at your publication history, I saw you came in hot, right? You had like 6 publications or something in as many years of just sequencing all the genomes of these sea species. And it just stuck out to me, or what bled through to me, and you can correct me, is how the framework of thinking, you had to try and like put together the biology of these organisms based on their genome. And that right there created a whole new kind of science and scientist, where you had to reconcile or rationalize the complexity of the genome and overlay it onto their biology. And here you are now, like incorporating that into this new model, which I think is a kind of an extension. So again, just a great illustration of this nonlinear path to science and discovery. So for the final question, before we get to a couple peripheral cues, I wanted to ask you to give your advice to trainees. I mean, in your lab, in your own lab, even having had your experience, you really have a unique insight into that nonlinear path. What's the advice you give to this next generation of trainees in finding their own path in science and discovery?
Yeah, I mean, what I tell my students is that they have to care about the questions. That, I basically say that, through our training, we can learn all the skills. Very few of us are actually doing rocket science, right? Like anyone can learn what I do, but what I can't teach people is curiosity and the drive to answer questions. And so, I think what I've done through my career, and that's been the joy of my experience, is to just go after things that seemed interesting. I did not worry so much about what the consequences of those choices would have been, right? I mean, certainly it could have ended badly, right? It could have been that none of the species I tried to get working in the lab might have worked, right? Um, I took risks driven, I guess I felt like I couldn't help but do what I was doing, and was just following my nose on what I thought was interesting. Some of it was actually a bit of a hipster attitude, where I'm like, well, I want to be doing something nobody else is doing. And one of the beautiful advantages of taking that approach is there's very little literature to read up on, right? Because if nobody's published on the species, you're the first one. And so, maybe partly it's because I don't like reading too many papers or something. But I think finding that questions that speak to you, I think, is important, because especially with graduate students, I remind them at the beginning of their careers that grad school will feel hard at some point or another. I don't know anyone for whom grad school was just this like straight, no problems type of experience. And so what gets you through those tough times, because there will be experiments that fail, there will be papers that get rejected. So I think what helps you go through those tough times is the questions that, something keeps you coming back to the lab. And so to me, keeping it questions focused was very valuable personally, and that's how I try to motivate trainees in my lab.
Yeah, that's a good way to put it. That's a good way to do it. Follow your heart or follow your nose, like you alluded to, like you mentioned. Science is tough. You mentioned, and we all have experienced it during our own training. Nothing is linear. A PhD is a very nonlinear path. Failure is normal. And I think the sooner the grad students can learn that, the better, and better off they'll be, I suppose. But, the work that you're doing is really taking that to the next level. And when you're developing an entire new model system, that takes an incredible leap of faith and a tremendous amount of courage to do that. So we applaud you for your continued efforts, and we can't wait to see what's going to come out of your lab in the years to come. Whether or not you go back to Bermuda, we'll see what happens. But before we let you go, we're going to ask you a couple of peripheral questions, like Daylon alluded to. First off, what's a hobby that you always wanted to pursue but were never able to because you're so busy?
I would love to play the cello someday. Unfortunately, I'm not. I don't have very good musical instincts. As in, I love listening, but I've tried learning how to read music. I've tried playing a few things here and there. And my 9-year-old just picked up the cello, and of course, I said, "Oh, maybe as she learns, I'll try to." But she's, zoomed far ahead. So my plan is that when I turn 50, I will start taking cello lessons. That's my new goal.
Those darn kids, they're kind of like those pluripotent stem cells, so malleable that they
Exactly.
can pick things up really quick. I mean, as the father of a 4-year-old, I can totally relate.
Beautiful analogy.
Absolutely. Last question. If you're not a scientist, what would you be?
I think I would be a linguist. I think, to me, again, like I said, I think what drives me when I look at the world is to think about how things got to be the way they are. And, linguistics is essentially evolutionary biology, but with words. So um, that has always been interesting to me.
Wow. I could see that. I mean, the interlocutor that you are for the worms and the humans, I think languages would be a cakewalk for you. As Arun said, I mean, this has been a delight having you on. And as someone who we've never had it before, someone who created the model. And it really was such a treat to hear you describe those little details. And quite honestly, I'm going to be honest here. I didn't realize that it was that tough. I was like, oh, here you go. You get some worms, you get them in the thing. What worms? They're alive. They make it. But no, there's all the unexpected challenges and all the optimization and all the application of all the new tools. I mean, because that's the new deal, right? With the models nowadays is they got to fit in with this amazing tool set we have. So that was really fun. And we really appreciate also your insight in terms of like mentorship and outreach. This has been the total package. Mansi, thank you so much for joining us on the show today. We hope to have you back on again soon.
Thank you. And if I could just jump in with, it's, I think, easy to interview one PI and think that they did everything. It's a perspective that I really try to correct. Remember, it does take a village. A model system is not born by, one person doesn't give birth to it. It really is. I've been super privileged in having truly incredible and gifted students and postdocs come through the lab who have given the work their all, and they themselves have taken huge risks, right? It could have been that transgenics didn't work. It could have been that the embryos would die when you pull the cells apart. But people came in willing to try things and actually proposing new ideas of their own. And so it's been a real fun time.
Well, thank you for sharing that. We cannot emphasize enough on this show that the trainees are the real engine that's driving this effort. And in your case, yes, taking all those same risks. But thankfully, they have you as a leader, and maybe there'll be a trip to Bermuda in there at some point. Remains to be seen. Anyway, thanks again. We had a great time chatting.
Thank you.
All right, guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, or you can hit us via email at info@stemcellpodcast.com with feedback or to suggest guests. We also have a little in-betweener episode for you next week, so we'll be back in your ears before you know it. But until then, thank you so much